Λοιπόν, κυρίες και κύριοι συνάδελφοι, καλησπέρα. Όπως θα έχετε προσέξει, έχουμε αλλάξει την ώρα, γιατί έγινε κατόπιν αιτήματος πολλών γιατρών ότι θέλουν να είναι στη 1:30 για να έχουν τελειώσει τις δουλειές τους και να είναι μέσα στην αίθουσα. Βεβαίως, μην γελάτε, έτσι. Δυστυχώς. Και γι' αυτό το κάνουμε στη 1:30. Ελπίζω να έχει κόσμο και να φέτος στις επιστημονικές εκδηλώσεις και να έχουν και κάποιο νόημα, διότι ξέρετε και ο ομιλητής όταν μιλάει σε άδειο ακροατήριο είναι λίγο άσχημο. Τέλος πάντων, σήμερα λοιπόν έχουμε την χαρά, και πραγματικά χαρά είναι, να έχουμε μαζί μας τον κύριο Κόλλια, Σπύρο τον Κόλλια, καθηγητής Ακτινολογίας και Νευροακτινολογίας στο Πανεπιστήμιο της Ζηρίχης, και ο οποίος είναι πολύ γνωστός για τα θέματα αυτά που τον αφορούν όσον αφορά την νευροακτινολογία, και σήμερα θα μας μιλήσει για ένα θέμα που είναι πάρα πολύ ενδιαφέρον: ενόραση στη δομή και τη λειτουργία του εγκεφάλου. Παρακαλώ, κύριε Κόλλια.
Είναι χαρά μου να είμαι εδώ. Την ομιλία θα την κάνω στα αγγλικά. The first insights into the structure of the human brain came approximately at 1700 to 1400 BC from the ancient Egyptians, that they were observing people with traumatic brain injuries and after human sacrifices. I will move to 400 BC, όπου ο Ιπποκράτης was the first one to recognize that there is a cleft between the two cerebral hemispheres. Later on, Platon believed and introduced that the center of intelligence was the brain, in contradiction to the beliefs of Aristotle, who believed that all the feeling, all the sensations, the intelligence is in the heart. This is how it started, the noocentric versus the cardiocentric different groups in medicine. With the advancements of computer science and imaging techniques, the investigation of the brain through neuroimaging, it became a very important part of the neuroscience disciplines. Essentially, neuroimaging started in 1972 by Godfrey Hounsfield, who introduced the X-ray computed tomography. Based on some physical principles related to the way that water protons behave in a magnetic field, that they were introduced by Bloch and Parcel independently and prolonged to the imaging by Lauterbur much later in 1973, has been introduced nuclear magnetic resonance in the imaging of biological tissues. And we come only in 1980, where Hawkes, Holland and Moore, three physicists, together with one neuroradiologist, Worthington, they demonstrated for the first time an intracranial pathology, a tumor, using MR technology. The advances of structural neuroimaging and of the technique of MRI rely on the high spatial and temporal resolution of the technique, the possibility to sample the anatomy in different planes, the very reliable differentiation between normal and abnormal tissue and between gray and white matter, with or without contrast administration, the possibility to obtain non-invasively angiographic images, and predominantly the lack of irradiation hazards made this modality the modality of choice for understanding human neuroanatomy and evaluation of CNS pathology. Here is an image from the 70s, a CT image, which demonstrates one of the first images that demonstrates an intracranial pathology. And here we have a movie from what we obtain today with an acquisition time of 3 to 4 minutes. We can generate images, three-dimensional images, and study the surface of the brain with the multiple foldings, the solci and the gyri. Then we can dissect through the brain, study the peripheral neuronal populations forming the cortex, the deep neuronal populations that they form the neuronal populations arranged around the ventricular system, and the white matter in between connecting the different neuronal populations. So a dramatic change within a very short time of evolution of neuroimaging. The non-invasiveness of the technique, it allows, sorry, here we had a video but it doesn't work, it allows study in the intrauterine phase. It allows us to study the anatomy and the development of the brain. Here we see a brain that looks lysencephalic. However, this is completely normal. It's from a 7-month-old fetus. Here we see a profoundly abnormal brain in a child with a cortical malformation, a band heterotopia with devastating clinical consequence for the child. It revolutionized surgical approaches, particularly in the field of neurosurgery and in the field of head and neck surgery. And this was because it showed very clearly and very precise the location of the pathology in relation with the important anatomical structures of the brain. We have a patient here with an abscess in the occipital lobe. We see the three-dimensional images, and here we see the evacuation of the abscess, which was done interactively with MRI imaging guidance. It also revolutionized, and that's why we have already 3 Nobels in the field of neuroimaging, it revolutionized the way public looks about neurodegenerative and mental processes and mental illness. Here we have a coronal image from a small structure on the one and the other side, which is the hippocampus, normal subject. Here we have a profoundly abnormal case. We see that one hippocampal formation is very small in a child with intractable epilepsy. If we go back 50 years, when the first studies using computed tomography were published showing that patients with schizophrenia, they had an enlarged ventricular system and atrophy of the frontal lobes, this was absolutely the turning point where schizophrenia does not anymore belong to a curse from the God, but to a neurobiological illness. And what it means, neurobiological illness? It means that it is something that we can fight, we can look for treatment, and we can improve the life of the patient and not close him in some mental institution. Very soon it was realized that the signal that we get from a tissue, from a volume of tissue with MR imaging, it is not only sensitive to the water content of the tissue, but also to several other biophysical and biochemical properties that they affect the relaxation characteristics of the specific tissue, such as temperature, viscosity, tissue microstructure, blood flow and blood volume, water diffusion, blood oxygenation. They can be manipulated to alter the relaxation characteristics and therefore the image, the signal intensity of the image that we obtain. Therefore, it was suggested that these alterations, They can provide information on physiological, biochemical, and functional processes in the brain. So, as a first definition, functional neuroimaging is a MAR technique that allows evaluation of regional or global cerebral physiological processes. And in the present day, we can study cerebral blood flow and tissue perfusion using the techniques of MR angiography and perfusion MRI. We can study molecular brain structure and myeloarchitectonics using diffusion MRI and diffusion tensor MRI. We can get insight into blood-brain barrier permeability by doing contrast-enhanced studies. We can study regional neuronal activity using the so-called BOLD fMRI, and also we can study brain metabolism using MR spectroscopy. And we will see some of these applications, since our time is relatively short. So, first insights into the vessel flow and perfusion physiology of the brain. As we know, blood flows in the brain in a rate of approximately 0.5 to 2.5 ml per gram tissue per minute, supplying the brain with oxygen and nutrients through the arterial system and evacuating the byproducts of the brain metabolism. They have been developed several techniques which emphasize the arterial flow, like this TOF angiography that we see here, that we can see the primary, secondary, tertiary branches, arterial branches of the intracranial circulation, or they emphasize the venous flow with several applications in clinical medicine. Until approximately 25-30 years ago, MR angiography provided non-invasively, without any contrast, provided only static images, although flow information was contained within the acquisition. So here we have an example from a vascular pathology. You can recognize it, most of you. We have a patient, we have the anterior cerebral artery, middle cerebral, internal carotids with a huge aneurysm in the anterior communicating artery. It is a disease, it is a pathologic process which is not uncommon. If we were 100 people here in the room, which we are not, 5 of us, we would have an aneurysm. Some of them, they are going to rupture, some of them, they are going to remain silent. If they rupture, they have a relatively dismal prognosis for neurological outcome. This is a static image. So we have the information of flow from the static image, but we can also extract from the MRI the velocity information. And this way, by collaborating with very competent people from the computational physics, in this case it was my collaboration with the ETH of Zurich, we can move from the static image to an image that it gives us a much more information than we get with the static image. So we have the anatomical frame, we see the pathology, we see it three-dimensional, and then using computational simulations, we can, in the individual patient, create accurately the flow streamlines that they are quite stream in the parent vessel, and then we see the chaotic flow inside the aneurysm, and this is a quantitative information that it can give us additional insight into parameters like shear stress, wall stress of the aneurysm, and therefore to start gaining insight into the fragility of the aneurysm, so to separate which aneurysms they are going to rupture and which ones they are not going to rupture. If you have a computational model with realistic 3D anatomical information and physiological information, You can slowly add additional parameters. You can add humoral parameters, chemical parameters, parameters related to blood pressure, and therefore test the model and test the fragility of the specific pathology, of the specific aneurysm. And of course, changing each parameter every time. Only one parameter. If you change all the parameters together, you are just going to be in a chaos. Magnetic resonance, it can provide, as I said, information on the secondary and the tertiary branches of the arterial tree and of the venous. However, we cannot get inside due to the limitations in the resolution in the microscopic angioarchitectonics of the human brain. Only with high field systems we start getting first glimpses of the small vessels that they supply the cortex and the white matter. So how we can use this technique to get information into the perfusion of the brain? Let's do a very small experiment here, very quick experiment. Here we have one slice in the brain, and suddenly I will give some contrast. So the first slices that we will see are slices without contrast, and then when the contrast goes through the arterial tree, we see that the signal intensity of the image, it goes down. This is the signal intensity versus time plot that we have here, and this, using equation of indicator dilution theory, it can be transformed into concentration versus time, so we can get information about the concentration of blood flow within the brain parenchyma. And then we can generate parametric maps. These now are not anatomical images, these are functional images of different hemodynamic parameters related to regional cerebral blood flow, regional cerebral blood volume, and mean transit time. Clinical applications, a lot and very important. Neovascularity and hypervascularity, as you know, it is a surrogate marker of malignancy. The more malignant is tumor, the higher the neovascular network that it has. When you perform a biopsy, you want your needle to go in the area of the highest malignancy so that you can plan the appropriate treatment for the patient. Anatomical images, very blurred image, even my 6-year-old can see that this brain is not normal. This is the post-contrast, we don't see a particular contrast, and here we have the cerebral blood volume, and from all this mass we can identify an area in the medial part of the tumor which has the highest perfusion due to the presence of the neovascularity. And then with MR guidance we can go with a biopsy needle and biopsy exactly the area that it showed the perfusion that it is the most malignant area of the tumor. Now, how can we use this technique? We will move now to another example, another application. How we can use this technique of MRI, which has a resolution of millimeters, to get insight into the microscopic organization of the brain. For this, we have the technique of molecular diffusion. As we know, the brain, it is composed essentially by two types of cells. These are the neurons and they are the kinks. We have approximately 100 billion of them. They receive, store and transmit information to neighboring and distant neurons. And we have the glial cells. These are the slaves. We have approximately 1 trillion of them. Some of the glial cells, the so-called oligodendrocytes, they will form insulations around the axons of the neurons so that the information it is transmitted faster and most effectively. Other ones, the astrocytes, they support the different metabolic functions of the neurons. So we have the neurons, the important ones, and the glial cells. Now, what is diffusion MRI? Water protons, because essentially with this technology we image water protons. Water protons, they move in any tissue, including the brain tissue, with approximately 17 microns in 50 milliseconds. During this movement, they bounce in the different components of the tissue, like cell membranes, myelinated axons, other macromolecules, which they alter the speed with which the water protons move within the tissue. So molecular diffusion, it comes from the Brownian motion, which is due to the thermal energy of the water protons, and it is random in any direction. It is expressed with a coefficient, the so-called apparent diffusion coefficient, which essentially gives us information about the structural integrity of the tissue. It depends on intra-extracellular water homeostasis and ionic equilibrium. One disease process that affects profoundly the intra-extracellular water homeostasis and ionic equilibrium is the arterial ischemia. And here we have an example from a patient with a stroke, with an arterial ischemia. We have the conventional images, T2, they don't provide very clear information. It needs some experience to see that here we have beginning of an edema. We have the angiography which shows the occlusion of the middle cerebral artery. The pathology is very clear in the diffusion MRI and in the apparent diffusion coefficient map. We have a failure of the potassium-sodium pump. We have accumulation of sodium intracellular followed by water, and we have cytotoxic edema. The cell swells, the extracellular space becomes smaller, which means that the movement of the water protons is restricted, and therefore we have diffusion restriction, which we can see in these images that they are not as clear as the anatomical images, but they show us very clear the pathology. If we combine the diffusion images with the perfusion images, the parametric maps that we get from the perfusion, we can identify patients that they still have salvageable brain tissue and they will benefit from early treatment. The most exciting application of diffusion imaging is the so-called diffusion tensor imaging. Diffusion tensor is based on diffusion anisotropy. Diffusion is a three-dimensional process. Movement of the water protons may not be the same in all directions. It may be one direction that it may be faster than in the other two directions. This is because of the presence of obstacles that they restrain the movement of the water protons. What is highly anisotropic? The brain of the mammals and the spinal cord. Why? Because they have the myelin sheaths around the axons, so the water protons, they move faster along the axons than they move perpendicular to the axons. And the white matter, it is not just a blob of tissue as we see it in the conventional structural images. It is a highly organized structure with sheaths, with axons that they run parallel to each other, and we have three main types. We have the commissural system, goes from left to right, connecting homologs areas of the two hemispheres. We have the association system, it goes between different neuronal populations in the same hemisphere, and we have the projectional system, which projects information from the cortex down to the brainstem and eventually to the spinal cord. So it was obvious that we can exploit the signal that we get based on the anisotropic movement of the water protons to create images that they depict anatomically these connections in the white matter. Here we have an anatomical specimen. We see the commissural system of the brain callosum, and here we see the reconstruction of the commissural system using the MRI technique. Why this created so much excitement? Because these myelinated axons connect different neuronal populations, connect cortex with the executive system. It was the first time that we might have information related to human connectivity, in parentheses human behavior, using a non-invasive technique. I will show a neurosurgical application because I did a lot of them since I was a clinical neuroradiologist parallel to the research. So here we have a patient who has a very large tumor. It does not affect the Broca area. It does not affect the Wernicke area. The person had speech problems. It affects, though, the arcuate fasciculus, an important association fiber system which connects the Broca with the Wernicke area, and it can produce a form of aphasia. We show the anatomical we show the pathology, but also we show the relation of the tumor with these important anatomical structures, not only the functional, but also the connectional structures. And now let's move to the last example, and I will be finished soon. How we use this technique to get insight into the neuronal function of the brain. The brain, in its complexity, it has four main functions. It senses stimuli from outside, sound, sight, taste, touch. It processes the incoming signals. It stores some of these signals as internal representation and controls the actions of the human body by regulating the muscular, immune, endocrine, autonomic system. Classical neuroanatomists, they arrayed all these different structurally and functionally anatomical areas in the human brain, in the non-human primates, in approximately 50 different areas, cytoarchitectonically different one from the other and with different functions. And we are talking a long time ago, more than 100 years ago. If we consider that now there are much more areas that they are developed, that they are found in the brain of the non-human primates, but also the huge enlargement of the brain human cortex in comparison with the non-human cortex, these areas now they may be more than 150 different specialized functionally areas in our brain. How do we use the MRI to map and to show non-invasively where these areas are located? The physiology of neuronal activation, as you know, neuronal activation is an electrical signal. If you listen to it, it's tick, 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 tick. Okay, and we can register it. However, this electrical activity is followed by a hemodynamic epiphenomenon. We have local increases in the brain activity, and these, they don't only produce electrical signal, but they are accompanied by an increase in blood flow and glucose utilization by the neurons. This was demonstrated a long time ago by Fox and Reichle using PET technology. These flow changes exceed the oxygen consumption by the neurons. Do we know why? We don't still have a clear answer to why the brain during activation resorts to a relatively anaerobic metabolism. It gets much more oxygen than the oxygen that it uses. However, this difference means that we are going to have more oxygenated blood in the areas that they are activated. And this local excess of oxygen is detected as so-called blood oxygen level dependent signal using the functional MRI. There were much debates whether this hemodynamic epiphenomenon that we identify using MR technology, it corresponds really to the anatomical localization provided by electrophysiological techniques. Until it came this ingenious project of Nikos Logothetis, he's a Max Planck, actually he's retired now, Max Planck professor in Tübingen in Germany, who did an ingenious experiment. He stimulated the visual cortex of cats using a stroboscope, and he did fMRI, and at the same time he did also single cell recordings using the needles that the neurophysiologists use for single cell recordings. What he found, and it is shown in these diagrams here, it is that the bold signal, so the hemodynamic epiphenomenon, directly reflects the input and the intracortical processing of a stimulus by the neurons. In other words, the anatomic localization of function that we detect with fMRI, it is quite close, if not the same, with the one that the electrophysiologists detect using electrophysiological methods. And we demonstrated also this, for example, in patients with lesions affecting the visual cortex. We did fMRI, and we correlated the fMRI maps with Goldman's perimetry, and we found a very good correspondence between the visual field effects detected with Goldman's perimetry and the localization of function using the fMRI. We correlated also this with electrophysiology. Patient with a tumor in the precentral cortex, he could not move his right hand, and we correlated the area of activation with the electrode that corresponded to the motor cortex during electrocortical stimulation intraoperatively. The impact that fMRI had in neuroscience is huge. fMRI started at the late 90s. At this time, system neuroscience was a dead field, was dying. Nobody anymore was interested in system neuroscience. Everybody was interested in molecular neuroscience, in genomes, in prions, in whatever, but not in system neuroscience. At the present day, we have at least 20 new journals that they are devoted to system neuroscience and mapping, functional mapping of the human brain. And we have at least 1,000 publications every month that they come out using this technique. So it was a huge shift in the neuroscience community, not back to the system neuroscience, but not leaving the system neuroscience to die. The signals that we detect, they can be very small. Like, for example, in this case, here we have a patient who has tetraamelia, absent legs and hands, despite the fact that she was feeling actually the movement of the hands and of the feet. And doing fMRI, we can demonstrate the areas that activate the network, the motor network that activates during movement of the phantom limbs. To make the long story short, if you are interested, another time we can give a talk on fMRI or in any other of these techniques. There are two important things that we learned with fMRI that we haven't realized with electrophysiological studies. One thing is that any task requires several brain regions to work as one system. Even a single movement of the right hand does not activate one area, but activates a network of different areas in the precentral, premotor, premotor inferior, prefrontal, in the lateral part of the contralateral and of the ipsilateral hemisphere, and also in the internal surface of the brain. So a simple task, it does not depend only in one area. There is a network of areas that they activate during execution of a simple task. Another thing that we learn is that a single region may play a role in seemingly unconnected task-related systems. For example, here we have a subject making a word generation experiment. Generates words, for example, we start with A matching task, deciding whether something is the same or different. And someone who does a hand movement, they activate different areas in the brain, but all of them, unconnected tasks, they activate the inferior premotor cortex, and we see here the activation. So functional specialization, it's something that with the time it starts to be departed. We are not talking about functional specialization, we are talking and functional areas, we are talking about functional systems that they work together in order to produce behavior. This was a short, very short to what neuroimaging makes. I like to close this talk with these words from Edward Wilson, professor at Harvard, unfortunately died 2 years ago, and philosopher, who, when the microscopes were developed, he said, our search for the ultimate through direct visual observation by steady advances in the resolving power of neuroimaging satisfies an elemental craving to see all the world with our own eyes. Σας ευχαριστώ.